mostly already made. Tonight is The Power of iNaturalist with Brandon Wu. 2 weeks from tonight will be me talking about fire in ways that you maybe have not thought about fire before. 2 weeks after that will be Dr. Glenn Dolphin talking about Alfred Wegener and continental drift. Then we'll take a break for December 31st for New Year's Eve, and then we'll have Dr. David Margolius talking about the science of COVID-19. And before Brandon starts, Rob Ross, my colleague at PRI. Oh, and I should also say that I'm Don Haas, the Director of Teacher Programming at the Paleontological Research Institution, and we are your host for Science in the Virtual Pub, and welcome one and all. We'll also ask that you keep yourself muted during the chat, and I'll keep, or during the talk rather, and I will do my best to keep an eye on the chat and see if there are pressing questions. I'll raise them to Brandon, but he doesn't need to pay attention to the chat. And before Brandon takes off, I'm going to, Rob Ross is going to say a few words about our BioBlitz program, and Alex Moore and Ingrid Zabel are both on and both involved in that as well, so they might chime in as well, but I'm going to hand it off to Rob.
Okay, so yes, I hope that Ingrid and Alex will chime in with things that I miss. I'm going to be very quick, but I'm going to share my screen and just say a few words to give you some context about how iNaturalist is related to the Paleontological Research Institution. I don't know if that even occurred to anybody, but there may be people on in this Zoom meeting that don't realize that we have these different parts to PRI. We have a natural history museum called the Museum of the Earth, and we also run a nature center called the Cayuga Nature Center, and we've been running some BioBlitzes associated with the Nature Center. But then you might say, wait a second, what does a Paleontological Research Institution have to do with the Nature Center or with biodiversity for that matter? So I just wanted to say a few words about that. So first of all, if you come to the Museum of the Earth, you'll take a walk through the history of the Earth and its life. And as you can see from the title, paleontology is the original focus of the organization when it was founded back in 1932. And we run one of the largest research collections of fossils in North America, and with something like 7 million specimens, or maybe that's wrong. It seems to grow all the time. And so you'll see a tiny fraction of that. There's about 700 or 800 of them on display in our museum. That is a tiny fraction of what's in our collections, but the collections themselves represent a tiny fraction of the biodiversity through the history of the Earth. And part of what paleontology does is try to understand why life waxes and wanes the way it does. And so I'm just going to show one plot. This is probably the most famous plot in paleontology. No worries if you haven't seen it, but this is a plot of the history of biodiversity through time in the oceans. So on the left, starting at 0, of course, and this is just animals in the ocean, and then gradually climbing toward the right, and this is today. And you'll notice there are some steep drop-offs in certain places where there are mass extinctions. And these have clear implications for what's going on today. And so there's a lot of interest in trying to understand, why do groups of organisms disappear from the Earth at certain intervals of time? And some, of course, there is this sort of constant background extinction, we call it, but there are also times when lots of things go extinct in a relatively short interval of time. And that's happening right now. We want to understand what is happening to biodiversity on Earth right now. How is it being impacted by climate change and other kinds of local environmental changes, invasive species, and so on. So although paleontology and forests paleontology of marine organisms and living forests might not seem closely connected. From our perspective, they are all part of the same thing. And so we think about all of those things. And so PRI has several campuses near Ithaca. One has the research collections and the museum. This is for people that aren't local. So, but we also have this separately. We have this nature center that has about 100 acres of forest and some other kinds of habitats. And we also have a 30-acre patch of old growth forest. So these are trees that, as far as we know, have never been harvested, which is very unusual in the Northeast. And so it's the grounds on the Nature Center and Smith Woods that have been the focus of our first BioBlitzes, but we're interested really not just in these places. These are sort of model places for us to do things and help people understand about their local environments, and for us to be able to track environmental changes and biodiversity changes literally in our own neck of the woods. But we are really interested in having everyone participate around Central New York, in our urban environment, and you all, no matter where you may happen to be. And so that's what Brandon's going to be talking about, is one tool for doing that. So PRI has this very sort of complicated looking mission statement, but you'll notice that it's not just about the geological history of the Earth or just about fossils. It's about understanding the Earth and its life. And part of that mission is about stewardship. And so the things that we do to look back at the past are intended to be important for understanding the present and the future. Before we started doing iNaturalist, we were publishing guides to some of the local natural history. So we have a field guide to the Cayuga Lake region, and we have also a whole book about Smith Woods that's about the history and care and significance of Smith Woods. And the field guide itself is what you might think. It's basically focuses on the flora and fauna that's relatively local. So if you're looking for something that you can hold in your hands, and that shows you the most common things in our area, you should know about that. iNaturalist is this incredible tool that Brandon will tell you about, for taking it a step further to document the biodiversity around you. So we have been doing, we have done several BioBlitzes, and thanks especially to Alex Moore and Ingrid Zabel, who have really organized and been the forces behind these. The next one will be at the next Cayuga Nature Center BioBlitz will be this coming January, at the end of January. The ones we've run up till now have been 24-hour BioBlitzes. So from 10 a.m. on a Saturday to 10 a.m. on a Sunday. And then we are going to run another one in the spring, basically in celebration of Earth Day, that is part of a bigger effort called the City Nature Challenge BioBlitz. And we will still be taking observations, not just us, but through all the people that participate from throughout Central New York. But we are hoping to be able to bring as many people from downtown Ithaca, maybe other urban centers, into this as possible, because they are in many respects underrepresented in these kinds of surveys, but there's so much interesting nature that goes on every place, as you may know. So that will be April 30th to May 3rd. And now I just want to ask Alex and Ingrid, what did I forget? And I'll stop.
Covered it well, Rob.
Yeah, I think that's good. We want to encourage everybody to participate, and particularly after you've heard Brandon talk about how to get started, jump right in and join us. That would be awesome.
Okay, so we will then hand it over to you, Brandon.
Yes, take it away, Brandon Wu, on the power of iNaturalist.
Okay, let's see. I gotta start screen sharing here. We'll do this, share. All right, how's this looking for everyone? present. Okay. So, as Don said, I'm Brandon Wu. I am going to tell you about the power of iNaturalist. So first, a quick bit about me, so you quickly know who I am. I graduated from Cornell this spring in May, and I'm currently on a gap year, this is probably about the best year to have taken a gap year that I could have picked during a pandemic. And I've always been interested in nature, and basically always been interested in insects and biodiversity generally. And over the years, I also got into photography, and most of the pictures you'll see throughout this are mine. Like these photos on here are all mine. And I guess professional interests center mostly around taxonomy of Orthoptera, grasshoppers, crickets, and katydids, which are these guys right here. And to that end, I'll be going to Texas A&M next fall in 2021 to be a grad student working on taxonomy of Orthoptera with Dr. Hojin Song, who has the only lab in the US that does such a thing. So it's pretty cool. And so it makes sense with me being into nature and being into photography that I really just love online citizen science platforms. And I've been involved over the years with several different ones. Maryland Biodiversity Project I joined back in 2013, which is attempting to catalog all the living things of Maryland, and so I joined there. I submitted all my photos of things that I took pictures of in Maryland back when I lived there and when I took trips there. And nowadays I mostly help them curate Orthoptera observations and their pages. And I just joined eBird this year, which is out of the Cornell Lab of Ornithology. And I've really enjoyed getting to know that platform this year. That's focused on accumulating bird observations worldwide. And then the one I've been involved with the longest is actually Bug Guide, which I joined back in 2010, which is, so long ago now. But that's focused on identification and natural history of insects and other arthropods from America north of Mexico. So, and then today we'll be talking about iNaturalist, which I only joined in 2016. So what is iNaturalist? For those of you who may not know, I assume many of you do, but basically what it is it's a global citizen science project, and it's global. So which means all those other ones I was mentioning are kind of, have a more narrow focus on, documenting biodiversity of a particular location of a particular group of organisms. But iNat is global, so you can submit a picture of anything from anywhere on the globe. And the main purpose of iNat is aimed at getting people really outside and just kind of interacting with the natural world. And it's fairly simple. Basically, just people submit their observations, their photos, and or sound recordings of things that may be calling, crickets, birds, and such. And then there's just a community of people that work together to ID each observation, and which is helped by this computer vision AI, artificial intelligence, that they have developed at iNaturalist. And this is startlingly good with identifying photos, at least down to a higher taxonomic level like family or genus. And also, just to be clear, I do not work for iNaturalist. I'm not affiliated with iNaturalist. I just like it a lot. I find a lot of value in it. And oh God, how do I go forward here? This is not letting me go forward. There we go. So, and again, I'll go fairly quickly through this. It's fairly easy to use. There's a mobile app which is available on Android and Apple, and that's pretty easy to use. And you can also upload photos from the website. And so basically how it works is you just take a picture of something. I'm using this example of this grasshopper I just photographed about a week ago down near Buttermilk Falls before the snow killed them all. And you upload a picture, and the computer vision, the AI, will give you this list of suggested species based on what is most visually similar, and it'll also mention what is seen nearby. And most of the time it will also come up with this, we're pretty sure it's in the, and that can sometimes be genus or family or order. And I remember hearing this, when it gives you this, we're pretty sure it's in the, I think that's like 90% accurate, I think, from a talk that one of the developers gave that I watched. So that's really nice. And then these species suggestions, they vary. Sometimes they are accurate and sometimes they are not. And then what you can do is, if you know what it is already, like I knew this is red-legged grasshopper already, so you can just tap the species and submit it. And if I didn't know what this was, you can just submit it at the genus level, or if I didn't trust any of those suggestions, you can just type in like insecta or arthropod or whatever, and then you can leave it at that if you're really not sure. And then you submit it, and then ideally people come along and help you ID it and correct you if you're wrong, or agree with you if they are agreeing with you. And you can look on the website here, and you can look at your observations. You'll see the photo, you'll see when it was observed, and then it'll get the coordinates here, and you can see it on the map, the big red point being where your observation is, and then all the other ones being other observations of that taxon. So this is kind of you can use this as like a proxy range map. So you can see, okay, red-legged grasshopper has been observed a whole bunch of other times in the Ithaca area, which makes sense as a very common species here. And this is super helpful because you can see if there were no other red points in the area, or if the only closest red points were in Florida, then okay, maybe you're wrong. Either, and there's a couple of possibilities. Either you're wrong, or the AI doesn't know what it's talking about, or maybe you found something interesting. Maybe you found something way outside of its normal range. Maybe you found a previously undescribed population. Maybe you found just something that the AI didn't recognize. And you might think, okay, how often does that really happen? But let me tell you, it happens a lot more than you think. And I'll run through a couple of examples of that here. Okay, and then actually, before I go through those examples, I was just going to say I'm going to go through this explaining kind of how I use iNat as a citizen, and also how I use it as a scientist, because as you can see, this kind of opens up a world of possibilities of tracking data and submitting and learning about the world. And also, I forgot to mention that if these observations become research grade if I think 2 people agree that it's X taxon, which sometimes that leads to some issues. If 2 people agree on something and it ends up being wrong, then it's like research grade, but it's wrong. But I have to say any mistakes that happen like that are usually corrected pretty quickly by either someone with expertise coming in and saying, oh, actually it's this, or oh, you can't identify it past family with this picture. And so usually the data quality is pretty good. So as a citizen, I use iNat quite a bit. I take a lot of pictures, especially this year with quarantine, where I've had nothing else to do but go outside and look at stuff. I have been taking a lot of pictures and uploading them all. And this has been really helpful for me for a number of reasons, and for anyone, I think, because you can look at, you can pull up a map like this and you can see, where all your observations are. So it's kind of fun. You can track where you've gone and what you've seen in different places. I think that's a lot of fun. And then it also helps you learn about things, because the AI, for instance, is very, very good with flowering plants. It's very good with birds, reptiles, amphibians, butterflies, a lot of charismatic things like that. And those are groups that are outside my area of expertise of grasshoppers. And so this has helped me learn quite a few different plants and, fungi and birds and things that I, would have had to labor over trying to learn beforehand, and probably would have come to a wrong answer anyway. But now I can get a suggestion from the AI in minutes, and then oftentimes submit, and then have other people come in to either correct me if I'm wrong or confirm that, also sometimes within minutes, but obviously varying over days to weeks to months sometimes, if it takes the right expert to come on and see your photo. But I've learned a ton using iNat, and I've been able to track a lot of where I see different things. And so I think anyone can do this, and I think this is really fun and really powerful. And as a scientist, there are a lot of options here. You can look at a lot of these. In my case, I like Orthoptera, and I am very interested in just seeing the distribution patterns of these species, and so to that end, I spend a lot of time on iNaturalist, probably more than I should, identifying people's pictures. And there's an identify tab which is really helpful to just be able to filter by, say you just want to see Orthoptera from Tompkins County, New York, and only image observations with photos or only ones with sound recordings and various other filters. And then you can kind of just very quickly scroll through the observations and kind of agree with the ones that you know, or correct the ones that are wrong, or add IDs to the ones that are at a lower level, like insects. And so this is really, really helpful, and it's fun for me, but also just interesting to see what things pop up, and if things are going, or things are showing up, you know, outside of their normal range, or if things that are, say, traditionally southern things are starting to pop up further and further north into New York, or things like that, which, you know, with climate change, things are, a lot of species are, or if things are starting to disappear, formerly common things are starting to be reported less and less by people. There are there's a lot of things you can look at here. And again, many of the time, it's just the usual, you know, red-legged grasshoppers and common species, but the weird things show up, and it's not just once in a while. The weird things show up pretty often, and I'll run through a few examples here that kind of illustrate a few points. So this was one that happened a few years ago. This photo was submitted of a cricket from Central Texas, and when I first saw this photo, I was just like, what the heck is this? This doesn't match any of the known, like any of the cricket species that I knew from Texas. And so after digging through some literature, I determined that this had to be this Trigonodimymus belfragi, the Belfragis cricket, which is a very rare species that was previously known from only 4 specimens. And so I said so much in the comments, and we had a back and forth here, and the submitter was able to provide some extra photos that showed some of the other diagnostic features that I had kind of thought I'd been seeing in the first photo, but then I was able to confirm for sure, going back and forth with the original description. And this was super cool. Unfortunately, the specimen was not collected, but this did end up being the first live photo of the species, as far as I know, and it did get used on Singing Insects of North America, this website, which is a database of photos, range maps, and sound recordings of all the crickets and katydids north of Mexico. And of course, this species doesn't have any sound recordings because, you know, song unknown, of course, because it was only known from 4 specimens, and, you know, habitat unknown. Yeah, there's still very little known about this species. But, you know, here was a live photo of it, and, you know, the submitter has, you know, continued to be on the lookout for these. He has not found any more since then, but it's a little piece of data, one more piece of data, I think is useful, and I think that's pretty cool. But this was an example where I didn't get to see that the specimen was not collected, and I didn't get to personally see it. It's a lot more fun when I get to personally see the thing, this is an example of that. This is Phaneroptera nana, the Mediterranean katydid. Is a very common, like, yard and garden species in most of Europe, and it's sporadically introduced in North America. There's a few populations in the cities on coastal California, and then also in New York City. So kind of a weird distribution pattern, but it's kind of persisted in those areas for quite a few years now. And then back in 2017, this photo was posted from Cambridge, Mass. And I saw that, immediately recognized what it was, and I was like, oh my God, I think that's the first record from Massachusetts. And I said to the submitter, and he just got this at his backyard porch light setup. And I said, if you see another one, you know, it'd be great to grab it so we could get a specimen in a major collection to document that it's not only in New York City now. And sure enough, within, I think, a week or so, this guy actually had another one at his light, and I went down to Cambridge and met up with him. He gave me the katydid, and, you know, it's nice to meet other iNatters. You know, there's a lot of people that you see their pictures posted a lot, and then you actually meet them in real life. It's kind of fun. People that are out there just looking at things in their backyards even. And so I was able to have the specimen, and I was able to get some nice detailed shots of it. Here are my photos of the individual he was able to catch for me, and I got a couple pictures of the external genitalia, or terminalia, which are important for a lot of these species. And so we had some nice pictures of it, and then the specimen now resides in the Cornell University insect collection, and so now there is a vouchered specimen to show that this thing is in Cambridge. And since then, actually, the the same submitter, and I think a few other people have increasingly reported it this year and the last year or so, spreading in Cambridge, not just in the one guy's yard, but in several people's yards and some other parks in Cambridge. So it looks like it's here to stay. But it was first found in Boston, or in Massachusetts, on iNat. And so, again, pretty cool. And then another similar example of something that just happened.
There's a question on the difference in the genitalia, but in those photos. So.
Let me run back there. This is actually just the same insect. This one right here is basically the butt end from the dorsal view, and then this is the same butt from a lateral view. So it's the same insect. It's just kind of different views of different structures there.
Okay.
Makes sense. Okay.
And another question:
sorry.
what you're using for taking your pictures?
So I have a Canon EOS Rebel with a 100mm macro, and I have an off-camera flash. And so basically, what I have this white box. Actually, I have it right here. I can show you. I don't know if anyone can see this, but I have this kind of white box thing that I put the flash in there, and facing upward, the flash bounces off the top of the white box and diffuses down and gets you this nice diffused lighting. And for the white background here, I basically just get a piece of white printer paper and put the bug on it. And then it's just a matter of making the bug stand still, which is probably the hardest part of it all. Just corralling them over and over as they fly away and try to jump off the table, and but eventually it's just patience. Eventually you get them to stand still for a split second, then you fire off 100 photos and one of them will come out.
Do you, just to interject, do you use like the action mode on your Rebel?
I've just been using manual. I'm just clicking away.
Okay, just curious.
No, yeah, it's there are other lots of strategies that I probably also could use, but this is just one that has worked.
Yeah,
for
there
me
are
in the
also
past.
many comments that the photos are beautiful.
So thank you. I'd like to think they're pretty okay. And okay. So let's see, where was I? Okay, so this is just running through another example that's so kind of similar. This happened this year. This photo was posted of a grasshopper nymph from Massachusetts, again, also in Massachusetts. And I immediately recognized this as a member of this genus, Schistocerca, the bird grasshoppers. And this was pretty unusual because it's mostly a southern genus. Only 2 species have ever been recorded in Massachusetts, one of which is just a vagrant of a very strong flying species, Schistocerca americana, that just occasionally shows up in Massachusetts just because it's a very strong flying animal. It gets blown northward and such. And this is a nymph, which indicates that whatever this was breeding there. And so I thought, well, okay, it can't be the vagrant one. So the only other option was Schistocerca lineata, lineata meaning lined, but it's the spotted bird grasshopper. Weird common name, but this is a species that's kind of pretty interesting. It's widely distributed across the US, but it just pops up in kind of isolated sporadic populations that often kind of look very different from each other in coloration. At least the details of the genitalia are basically the same. And then they also, and different populations that are isolated from each other are feeding on different plant species, which is kind of an interesting evolutionary puzzle. And and actually the guy I'm going to work with down in Texas A&M, Ho-Jin Song, is the one who has worked on these, who's kind of trying to investigate the evolution of this and other members of the genus. And so I figured that it had to be that. And so I said so much in the comments, and it happened that later in August, I happened to be in that area of Massachusetts, and since this had GPS coordinates, which most of these observations do with pretty reasonable accuracy, I was able to go right to the spot, and I found adults. The adults are maybe not quite as exciting. The nymphs are kind of nicer and green, but the adults are subtly beautiful, I think. And so males and females bigger here, males here, and then these are again 3 shots of the same males end of the abdomen, these different structures that are useful for in taxonomy. A lot of bug taxonomy is looking at the genitalia. But anyway, so I was able to find these things, and it was cool for me personally to see them, which is great. But I think more importantly, I mentioned this to Dr. Song at A&M, and he said he's collected these things all over the country, and he said he'd never seen populations from Massachusetts. And then I brought some of these things in to try to keep them alive and see what they were eating. And as far as I can tell, they are completely specialized on sweet fern, which is not a fern at all. It's a little woody ericaceous shrub, which is very aromatically scented. It's one of my favorite plants, actually. And these things would not eat anything else. Like they wouldn't eat lettuce. They wouldn't eat any other plants that I could throw in there for them. They just would refuse to eat anything else, but they gobble up the sweet fern. And then I also mentioned that to Dr. Song, and he said, yeah, I've never heard of a population of these things eating sweet fern. So, and if he didn't know about that, then this is pretty weird, because he's like the world authority on these. So I was able to collect some of these specimens, and I'm going to send them off to him, and that will be useful for his work, and they'll be used for some molecular work and trying to puzzle out the evolution of these things. And this is a population that he didn't have specimens from, that he didn't really know existed, and now we know. So it's kind of interesting to see the connections here. Oh, sorry, what?
Another question is, are grasshoppers generally doing well, or are they facing stress?
What do you mean, like in terms of species, like going extinct?
Well,
Because,
Ingrid asked a question. Ingrid, do you want to unmute yourself?
Yeah. I don't know exactly what I mean, but I mean, you hear about bees and other insects that are struggling for various reasons. Are grasshopper populations doing well, or?
I'm sorry, it depends on the grasshopper. You'll hear stories of like desert locust swarms in Africa, or Australian plague locust in Australia, and some of those species are doing quite well because they're more and more of their habitat is being created by crop fields and such. But overall, I think Orthoptera, the grasshoppers and crickets, are probably one of the most imperiled orders of insects, because many of them rely on native grasslands and prairies and those kind of habitats, and those are among the most threatened habitats because they tend to be the first ones targeted for agriculture and kind of bulldozed and destroyed. I think it's better known in Europe. Which I'm not terribly familiar with, but I know a lot, like some huge proportion, like over 50% of Orthoptera in Europe are like approaching or some level of threatened or endangered. And I wouldn't be surprised if it's similar in the US, but I don't think people have studied them as much. But certainly a lot of the higher species diversity tends to be in prairies and other habitats. And as a lot of us probably know, the Great Plains and prairie habitats have all but disappeared under the corn and soy fields. So, but Orthoptera are overall kind of not doing great, but it's overshadowed by like some of the common species that are doing very well. And we kind of forget about all the other ones. If that answers your question.
Yes, thank you.
Yeah, and there's also a comment on the beauty of those lateral lines on the female.
Oh yeah, it's a lovely animal. So anyway, I was just kind of the point I'm making with this one is kind of these interconnectedness of somebody posting a picture of this grasshopper, me recognizing what it was, me being able to go there and collect the specimens, and then getting them off to Dr. Song, who will be able to use them in his research. And so there's all these different avenues for people finding things and then ending up being useful for somebody, one or two or three, lines removed here, and who might never have been able to come across such specimens otherwise. And you see these kind of examples all the time, and these are just some of the titles of some of the blog posts on the iNaturalist blog, where they kind of document a lot of these things that happen, and these are just a few examples that I just pulled. And you can see these span the gamut: westernmost record, first documented male-male combat, possibly undescribed spider, plant found hundreds of kilometers from its known range, first sighting of a rare cicada in a century. I mean, these things are just happening all the time, and across all different taxa and all over the world. And these are basically all, I just pulled them from 2020. Okay, this one's 2019, but these are just happening all over the world and in all different sorts of scenarios. And so and it makes sense because, with people out there taking pictures of things all across the world, all these cool things are going to show up. Not only just specimen wise of like, oh, that's a specimen we need for our research, but here was, a behavioral thing, first documented, behavior in the species, and all sorts of this. It opens up all sorts of possibilities. And so here's some of the screenshots of some of the bioblitzes that were put on by Cayuga Nature Center, PRI, and out in Smith Woods. And some of these things may turn up in bioblitzes. These odd finds may turn up at any point. And iNaturalist gives you the ability to make these projects of bioblitzes where you can kind of restrict observations to a certain geographic area and over a certain period of time or certain users. And it just gives you a lot of flexibility in being able to document biodiversity, either if you just go visit a place for one day and take a whole bunch of pictures, or if you visit the same place day after day after day and photograph the same plant over the whole course of the year as it, flowers and seeds and, dies off in the winter and then flowers again in the spring. And you can just you can track changes over years. So with that, basically, those are image credits, and I'd be kind of happy to talk about how any of you have used iNat and any other questions about using iNat. I've used it a bunch now, so if there's any questions about using it, hopefully I can answer them. Yeah, so I'm happy to open up the floor to talk about iNat. I'm a big fan of iNat.
That was great.
Actually, just one quick other thing. I think that, like I said before, the stated mission of iNat is not only to create research grade observations and research quality data, but also just to get people out there and interacting with the world. And I think that's a big part of it, because I think it's helped me quite a bit interacting with, plants and birds and fungi that I would not have maybe paid attention to as much in the past. And I think it gives people a better appreciation of things. It's not just, oh, let me go outside and look at a flower. It's like, oh, let me go outside and I'll take a picture of that flower and upload it to iNat, and that might be useful for somebody looking at that species of flower, tomorrow or 100 years into the future. This is creating a database of all these images and observations of different species in different locations at different times. And I think it's been really nice being able to connect. It's connecting people with nature in a way that I don't think has been done before.
Yeah, I agree.
Anyway,
I've gotten hooked on using it. So there are a couple of questions in the chat.
I can
three.
Stop sharing, I guess, if I need here.
Is there a way to pull images from other platforms and insert them into iNaturalist?
Oh God, let me bring this back here. Okay. Okay, a way to take images from other platforms. There is an uploader from Flickr, I think. So if you have a whole bunch of photos from Flickr, there's an uploader tool from there. Otherwise, I think it's just you can just cross-post them in terms of just uploading the same pictures there. There was one from Google Photos, but I think that is not working any longer.
Yeah, it's essentially, if you've got the picture, you can import it into.
Yeah, basically, if you have the file on your computer, you can upload it, and it'll have the date in the file already. It should, and then GPS coordinates. If you're taking it on your phone, you turn on GPS on your phone, and then it will pick up the coordinates of wherever you took that picture. And then if you don't have the coordinates, it's usually fairly easy to zoom in and find where you were. Like if you knew where you were at Buttermilk Falls State Park, you can kind of zoom in on Buttermilk Falls and then kind of make a circle with a reasonable accuracy over where you were.
And Mike asks, are you able to provide more data than just a photo and latitude and longitude, such as habitat or whatever?
Yeah, so there's a whole bunch. I mean, there's like comments under the photo, or details, I guess, under the photo, where you can add in all sorts of other data of habitat, or feeding on this plant, or called once and then flew to this plant, or whatever. And then I think there are also annotations on the side that you can add, like plant phenology, flowering, seeding, or adult, male, female, nymph, egg. So you can add on all those details as well, depending on what the organism is, whether it's going to be something that's flowering or has an egg or has a nymph.
And a question on your last slide that'll make you want to bring it back up, which is, Third picture from the left in the top row. What is that thing?
Bird picture in the top, that is a male American redstart.
Yeah, the third picture, the one next to that, which I'm pointing to, which of course.
Oh, the bug with the big
Yeah.
horn
Yep,
up
the
here.
bug.
Oh, I'm sorry. That's a
Yep.
that's a wide-footed tree hopper, Campbellentia latipes. That's actually fairly common around here. You find it on goldenrod and asters, and the whole bunch of them like to like stand together, and it looks like thorns. So you're about to like reach
Yeah.
for a stem, and it's like, oh god, there's thorns. Oh wait, it's just these bugs, and they're completely.
Can you
harmless.
say that one more time so I can write it down?
Oh, wide-footed tree hopper. There's a lot of
Thank you.
species of tree hoppers around, and
It looked like one, but I wasn't sure. I appreciate it.
yeah, there are a lot of species around here, and they all kind of have differently shaped horns. They're cute.
Okay, and Melissa asks, after my undergraduate studies, I could identify almost any native plant and tree, and then I moved to the West Coast and lost a lot of that knowledge. iNaturalist has helped me relearn the native species of New York. So I guess that's actually a comment more than a question. And I see a note to disregard an earlier one. But that was the question you just answered, so there was appreciation for answering that. And Ingrid notes you can also indicate if the organism you're observing is cultivated / captive versus wild.
Yes, and if the organism is captive or cultivated or otherwise not really wild, it'll push the observation to casual grade, so it won't show up on the maps. So it's like if you took a picture of some Asian plant in somebody's garden that is growing there, but it doesn't really occur in New York, say, then it won't show up on the map. So you won't have a map of like a whole bunch of points in like China and then one in New York, which is kind of nice. That's sometimes a way you can find misidentified things. I have a set of species sometimes I'll go through and where I know it's supposed to only be in Europe, and like grasshoppers say, and I know it's supposed to only be in Europe, and then I'll look at the map and I'll see like one in Texas and I'll be like, probably not. But if it was, that would be super cool. Sometimes like the Katie did from Massachusetts, sometimes things show up where they're not supposed to.
And Mike asks, how long before you get an ID?
Yeah, it depends on the person, and it depends on the quality of the photo, and it depends on the group of organisms. Like for a lot of things like birds, if you submit a picture of a bird people will ID it in like minutes. And as opposed to a lot of, if you submit a picture of like a small beetle or a small fly or something, that could take days to weeks to months.
And sometimes it's the AI does it pretty much instantaneously.
Yeah, and sometimes the AI can get things pretty instantaneously. And so yeah, it kind of depends on the group of like the difficulty in identification of the group of organisms and whether the right person is on there or not. So like for Orthoptera, at least, because that's what I know the best, there really wasn't anybody IDing grasshoppers from anywhere in the Northeast until I came on. And so there was just a whole bunch of photos. When I first joined, there was just a whole bunch of photos from like New York and New England that were just like at grasshoppers or crickets or whatever. And to me, they were obvious, but nobody that knew anything about grasshoppers had been on the site for a while. And that was kind of what prompted me to join, because it was getting to me. I was like, oh God, I need to help out.
Awesome. And then another question, has there been any optimistic results that you found with distribution of a certain species versus negative results?
Well, I guess optimistic results. I guess optimistic results in terms of
finding things that are good indicators of environment or that sort of thing. Yeah.
Let's see, I'm trying to think of any specific examples. I feel like it's definitely happened where I've seen photos of something that's kind of restricted to a certain habitat, and then I see a photo of it from particular places, and that it was like historically recorded from, but had not been seen recently, and then I'm like, oh, it's still there, and so there must still be patches of habitats for it. I can't think of any specific examples, but I know that's happened.
Mm-hmm. Yeah, I've been amazed by just how many different pollinators come to my goldenrod. It's amazing.
Goldenrod's a magnet.
least a dozen species of stuff I had never paid any attention to until I got iNaturalist. Then a question of, will it identify houseplants? I would assume that depends on the houseplant.
It depends on the house plant, yeah. Sometimes it will and sometimes it won't. And if it does, then somebody will usually knock it to the casual grade because it's obviously a cultivated thing and it's not really growing in that spot.
And do you know of any crowdsourced projects done with students that contribute to real research beyond just doing a survey of species in a local area?
With students, I mean, I know there are a lot of like college classes and things that use iNaturalist. A couple from Cornell actually, since going under quarantine, have used iNaturalist to document their findings for the class, or like do a virtual insect collection, or I think spider biology did virtual spider collections and things that way. But I guess that's not for real research. I'm sure it's been done, but I can't think of any examples.
And actually, I think the next comment answers that question from Gary. It's a middle school I worked with has included iNat in their science curriculum. The gym teachers use the data from the kids' pedometers. Interesting. And then Larry says, using the AI to give an observation the best ID you can also helps it get confirmed by other users. Some experts regularly search for new observations of the species they study. If there's no initial ID, they would probably never see the observation.
Sure.
Yeah, Don, I wanted to jump in and say that I posted pictures of garden weeds before I ripped them out this spring, and a woman from Illinois called me to send her seeds so that she could do the genetics on it.
Oh, that's interesting.
Awesome.
So I have a question. I'm just going to say it because I think it's easier than writing it. And it's probably not easy to know, but so when I like to see or watch people identify my stuff, basically. I leave a lot of stuff, maybe I'm lucky if I get it to like family or genus level, and I often don't trust the species level. And it's always exciting to me to see somebody get on and identify something. But I've been amazed at the randomness of it. Like sometimes somebody will get on and identify something at some kind of basic level that I posted a year and a half ago, and maybe there's just one or two observations by them, and I wonder, well, why did you choose to do that one as opposed to anything else I've left unidentified? And so I wondered, and I guess it's pretty clear that there are researchers out there looking for specific things and identifying them. But I think a lot of the things that are being identified on my behalf are being identified by people who are not necessarily researchers, but just interested iNaturalist people. And so I guess my question is, since I have not gone on to identify other people's stuff, what is the process by which, the average person who feels like they know enough to get on and identify things, like how do they know where to start, and what leads people to identify certain things but not others?
I mean, a lot of factors. I mean, sometimes I get what you're saying. Sometimes you just like have random people that seem to ID things, and you're like, who is this person? And then, I mean, you can click through and to their profile and see sometimes, and a lot of the times they will not write who they are. I think that's something I think that really helps on iNat. That's what I always do whenever I get an ID, is check and see who they are. Like sometimes they'll just say, sometimes they won't say anything, or sometimes they'll just be like, I'm an amateur interested in this, or sometimes they'll be like, I'm the world expert on this group of plants. But to take their ID with a grain of salt, depending. But I don't know. Some people, I feel like a lot of people join and are not quite sure where to find things to ID, and that identify tab that I mentioned is really helpful. But I think a lot of people miss that. like first time users kind of miss that, and they just go to the Explore tab and they just start seeing
Muted.
like all the observations, like just recently submitted observations, and then they just happen to click on like, oh, I know that's a chicory right there, and then they agree. So
I see.
I think
Okay.
people get a little confused sometimes, especially first time users.
Yeah, I find when I track down who's been identifying my things, they're usually people who say, yeah, that they sound like they're knowledgeable, but probably not researchers. Occasionally, I'll get somebody from a museum or something, and then it's exciting to know that maybe they're like really interested in following something on iNat for their research.
Yeah, and it always is kind of it's gratifying when you see something that's ID'd from someone that is associated with a museum or is often a recognized expert on such and such. You're like, oh, nice, exciting.
Okay, there aren't any more questions in the chat. Are there more questions? I posted a few links in the chat to PRI's homepage and to the page where you can donate if you appreciate joining us for Science in the Virtual Pub and our other programming, and the link to what's coming up in Science in the Virtual Pub. And as I said, 2 weeks from tonight, 2 weeks from tonight, we'll have me talking about fire in a different sort of way than you're used to. One bit of trivial I'll throw out as a tease is that, and I shouldn't say trivia, one interesting fact is that for 90% of Earth history, there was essentially no fire because almost all fire is burning biological material, and it was all wet until the Devonian period 400 million years ago. So that was a big change. And 2 weeks after that, we'll have Glenn Dolphin talking about Alfred Wegener and plate tectonics and a little bit of a biography on him and the mythology associated with him. And then we'll take a break for New Year's Eve. And on January 14th, we'll have Dr. David Margolius talking about the science of COVID-19. And a bunch of thank yous showing up there. And thank you very much, Brandon. It was a great talk and stunning pictures as well as interesting information. And any other closing questions or comments? Lots of thank yous popping up in the chat.
You're welcome, everyone. I guess the one thing I'd close with is, well, everyone submit things to iNat, and and if you. And again, it's very helpful if you just write something in your profile just to say who you are, because I find that helpful when seeing who IDs my pictures, and otherwise, just if you submit a grasshopper, I will see it. I keep track of grasshoppers and crickets from New York State daily. So.
Yeah, and I should also note that on the alternating Thursdays with Science in the Virtual Pub, we have our continuing discussions on reinventing education in the time of the pandemic. But a week from tonight is Thanksgiving, so we're taking a week off there, and so we'll resume that in December 10th will be the next reInvent Ed session, and the same link works for reInvent Ed or any Science in the Virtual Pub talk. So. Thank you one and all, and I think we'll wrap it up. And there is
Thank you.
This was on Facebook Live, and are both posted to our Facebook group for Science in the Virtual Pub as well as the PRI page. And if you want to share the recording with anybody, you can find it there. All righty. Have a good night, all.
Thanks, Don.
Okay.
Thanks, Don.
Bye-bye.